The topic for today, we have two things. First thing is I'm going to tell you guys a little bit about membrane lipids, okay? However, you guys should understand that a very big portion of fatty acids, okay, are used to make membranes, okay? So obviously, I think it was in Biochemistry I how there are 7 pillars of life, and one of the most important characteristics that define how you started life is to have a separation. Okay, so in order to have life, you need to have separation in space between your biological components and your environments. Okay, so that's important. Okay, so we're going to talk about membrane lipids. In particular, we focus on phospholipids and sphingomyelins. So this is a chart that's a very broad overview of membrane lipid biosynthesis. And what we see is that you have a difference between eukaryotic only, eukaryotic plus prokaryotics, okay, so the stuff that they share, and then eukaryotics only. Okay, so from this chart, you can probably already see how in eukaryotic systems you have more pathways, okay, more diversity in terms of how you get to your membrane lipids, okay? So from this overall pathway, let's first take a look. So from Biochem 1, we talked about several different types of phospholipids, okay, especially on the membrane, okay? And we saw that a lot of them are these phosphatidylserine, phosphatidylethanolamine, those type of stuff, and I'll show you the structure in just a minute. But then essentially you're asking what are their precursors, okay? So what makes these membrane lipids? Okay, so the question is, what makes these membrane lipids? Okay, so that's the question. So of course, we know that almost all membrane lipids, you have some kind of a hydrophilic head group, okay? And then you're going to attach it with 2 hydrophobic tails, okay? So in this case, what do you guys think the hydrophobic tails are? Hydrophobic tails 会是什么样子类型的 molecule? 大概 fatty acids, right? Okay, so these guys are going to be essentially fatty acids. And then you have a polar group on the top, okay? So, and I'll show you, it's really essentially, it's like glycerol, and here you have fatty acids, and then here where you put the head group. Now, there are different kinds of head groups, okay? There are almost all negatively charged or negatively charged plus positively charged. Okay, so different kinds of head groups, so this is what we're going to talk about today. Okay, so I'll show you where we get these things. Now, so you guys can understand that these 2 tails are coming from fatty acids. Okay, so as a result, in prokaryotic systems, okay, let's start from something very, very simple. Okay, in bacteria. Okay, bacteria. Bacteria, you first start with glycerol 3-phosphate. Okay, do you guys remember what glycerol 3-phosphate looks like? It looks like this, right? Glycerol, and then you have a PO3 minus like this, right? Glycerol 3-phosphate. And then you're going to add 2 acyl-CoA, okay? And remember, acyl-CoA are blah, 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 and then, okay, remember that's acyl-CoA. And your acyl-CoA here, it's already activated, okay? So then all you have to do is take this guy and take one of the OH on your glycerol or glycerol 3-phosphate. You react them, and you react them once, twice, okay? And then you'll get to this thing called the phosphatidic acid, okay, this guy, phosphatidic acid. And all this is essentially two arm, like two fatty acid arm, with a phosphate as the head group. Okay, so that's all it is. Okay, so very simple, right? So we go from glycerol 3-phosphate, this guy, into this by adding 2 fatty acyl-CoA. And where do you get fatty acyl-CoA? Okay, fatty acyl-CoA comes from fatty acid. Okay, here remember he used 2 ATP. Okay, it's kind of like in the degradation of fatty acids. Okay, and you get this fatty acyl-CoA, and once you hook up 2 of those onto glycerol 3-phosphate, you get this phosphatidic acid. Okay, and then it's going to get further activated by using CTP. Okay, and you form this thing called the CDP diacylglycerol. All it is now the head group has a CDP. Okay, because here these 2 tails, we can call them diacyl, right? So these 2 are the diacyl. And then on the top, the 3-carbon molecule is glycerol, right? So diacylglycerol. Okay, so diacylglycerol means basically this thing. Okay, so this is 1 acyl, 2 acyl, and then on the top it's glycerol. So this is diacylglycerol. Okay, now if it have CDP in front of diacylglycerol, then it's just a CDP attached to the head group and then diacylglycerol. Okay, simple. All right, now, so we know now your phosphatidic acid, the very basic, okay, 最 simple 的一种 membrane lipid 就是这个phosphatidic acid， 因为它这边有 2 tail 2个 fatty acid 的尾巴，然后再加上一个头是一个 phosphate，OK， 因为 phosphate 从哪里来？从 glycerol 3-phosphate，OK，phosphate 还在那边，OK， 所以如果这边是 phosphatidic acid 的话呢，它这边就会是一个 phosphate，OK， right， so now going to CDP diacylglycerol, you're okay. Okay, now what we're going to do is we can then synthesize a bunch of variety of different lipids, okay? And in particular, I want you to focus on this particular round, right? Those 3, okay? Most of our discussion will be focused on those 3. But you do have this other little thing where you take the CDP diacylglycerol, so we're looking on the right, and you react it with glycerol 3-phosphate, okay? And then you hydrolyze it, you get this phosphatidylglycerol, okay? And then essentially you hook up 2 of them together, you form this thing called cardiolipid, okay? But my emphasis will be first talking about the stuff on the left, okay? Because the stuff on the left are more commonly seen membrane lipids. Okay, so here's the actual pathways. We have the formation of CDP diacylglycerol, okay? So first, after forming phosphatidic acid, CTP donates CMP onto the phosphatidic acid, okay? Now, since phosphatidic acid already has 1 phosphate, together the CMP forms a CDP, okay? What does that mean, okay? All right, so again, we start with this thing on the top left, that's the glycerol 3-phosphate, okay? I'm looking at this guy. And glycerol 3-phosphate, again, is 1 carbon, 2 carbon, 3 carbon. Here's an OH, here's an OH, and here is a phosphate, okay? Now, glycerol 3-phosphate, okay, you take activated acyl-CoA, or in this case, here it shows you acyl-ACP, but pretty much the same thing. All right, so you take your activated fatty acid thiol ester, then you react it once, okay, and then you threw first arm onto the glycerol, okay? And then you do it again, and then you throw the second arm onto the glycerol, okay? Now, remember, the reason why this can react, okay, you can form an ester bond, is because here is a thiol ester. 记得哦，如果你今天是 fatty acid, 你没有办法直接做 fatty acid 加 OH, okay? You can't take fatty acid plus alcohol, react them spontaneously in biology. You can't, okay? Transesterification 我不知道你们有机化的那个实验有没有做过 ，OK？ 但是像我以前大学的时候，我们在做有机化 organic chemistry 的实验，我们就会拿不同的有机酸 carboxylic acid 加上不同的这个 alcohol，OK？ 然后我们要做 ester formation，OK？ 那 ester formation 呢？你们知道 ester 啊，其实是那个水果香精很多，就是它会闻起来会很像水果的味道。OK？ 那我们那时候在大学的时候做那个实验，刚开始前面5分钟很嗨啊，因为你就会一直闻到说、OK， 你的是香蕉的，然后我的是苹果的 ，OK， 就觉得还蛮酷的。但是当你做那个实验做2个小时，你整个实验室没有什么很通风的情况下，然后你吸那个东西吸了2个小时之后，每个人出去头都快炸掉，就是会头很痛。OK， 那ester formation 呢，在有机合成啊，你就是需要加热 ，OK， 加酸去催化。然后加热做 reflux， 你才能形成 ester。OK， now biology we can't do that。OK， 因为 biology 你不能莫名其妙的加热。OK， 那你也没有什么 acid catalysis。Right， so in biology, if you want to make an ester, okay, you absolutely have to first activate your acid. Okay, and by activating your acid, I mean, okay, you turn it into a thiol ester. Okay.Using the power of ATP, right? 从那个 organic acid 到一个 acyl CoA， 你需要花2个 ATP, OK？ 所以你是用 ATP 的能量去 compensate for 我们在有机合成的时候加的热能 ，OK？ Now, so you get this thing called the phosphatic acid, OK? I know it's a lot of names, but you just gotta know it. Phosphatic acid, it has 2 arms and 1 phosphate.Okay, and then what you're going to do is you're going to have an enzyme, okay, that's going to transfer the CMP, okay, it's going to transfer essentially this portion of the molecule, okay, onto the phosphorus, okay, so onto this phosphate, okay. And the way it works is actually very simple. You have lone pair, yeah, I mean not lone pair, you have electrons on the oxygen, it's going to attack the phosphor in the center, okay, and then pyrophosphate leaves, okay. Now, once you get the CDP glycerol, now it's even more activated molecule, and now we can do a little more stuff to it, okay. So CDP diacylglycerol is then used to form various membrane lipids, okay. So very briefly tell you, let's look at the right side, okay, very briefly. So you take CDP diacylglycerol and you take another molecule of glycerol 3-phosphate, you hook it on. Okay, so here this guy is the glycerol 3-phosphate. Okay, initially one of the OH reacts with the phosphate, the other OH is still here, and then you have a phosphate right here. Okay, so you used up one OH to react with your CDP. Okay, so you form a link right here. Okay, you form a link right there, and then okay, you get rid of your phosphate. Okay, you use a phosphatase. Okay, some kind of a phosphatase, and then you get rid of your phosphate, becomes hydroxyl group. Okay, and then you can then now you have 1 OH, 2 OH, and then you can add, of course, your phosphatidylglycerol. Okay, you add another glycerol, right? Essentially another one of this guy on the top, and then you have 1 tail, 2 tail, 3 tail, 4 tails. Okay, so you actually have 4 legs, and then you have a OH right here. Okay, so this OH is this guy. All right, so it's a very crazy lipid structure. Just to show you that it exists, okay? It does not account for a large percentage of your membrane, but it's there, okay? 哎，只是跟你讲说有这个东西啦，好不好？ Alright, now let's shift the focus on the panel on the left, okay? So let's shift focus to this part. You guys probably can't see it in the back, but whatever, okay? Okay, so we have CDP diacylglycerol. Now, okay, now, again, our goal is to have a membrane lipid. Membrane lipid has 2 lipid tails and 1 polar head group. Okay, now, essentially, all we're going to do right now is to change the head group. Okay, 只是 change head groups all. Right? Now, at the CDP diacylglycerol, the head group is what? Head group 是什么？在最上面 CDP diacylglycerol 的时候 head group 是什么？它的那个 CDP 嘛，对不对？就 CDP. Okay. All right. Now we're going to change that into some other moieties. Okay. Now, the first thing that we're going to do is we take serine. Okay. Serine is an amino acid. Okay. So you have serine, I have serine, everybody has serine. Okay. And you take serine. Now remember, serine's R group is what? 记不记得 serine 的 R group 是什么？啊，都在 slide 上了。只是你不要跟我说，老师我忘了 R group 是哪一个？那个有 NH2 的吗？不是嘛，那是 amino group， 对不对？还是那个 COO minus？ 不是嘛，那是 acid group。OK， 加在一起，它们叫做 amino acid， 有没有？哎，所以 R group 是什么？就剩下那一坨嘛。OK， 剩下那一坨东西，这个东西是 R group 嘛。OK， 那 serine 的 R group 是 simply a hydroxyl group. Okay, it's one carbon and then a hydroxyl group. Okay, now because your serine has a hydroxyl group, then essentially what you can do is you can have the serine OH react with the phosphate, and then the CMP can get kicked out. Okay, and it's a very, very simple substitution. Okay, the way it's going to work, obviously, okay, OH have lone pair electrons, okay, and lone pair electrons does what? It's going to, attack phosphor, okay, and then this extra bond here pops over, and then CMP is gone, okay. And then after you do that, and then you get this thing called the phosphatidylserine. Just like previously it was the phosphatidic acid, now it's phosphatidyl, okay, YL serine. Okay, 好，虽然你也不是很 care, but whatever. In short, you can call it PS, 不是 PlayStation, okay, 是 phosphatidylserine, okay. 所以这个应该很好记，就 PS 就好 ，okay? So this guy is PS, okay? And S obviously stands for serine, okay? PS. Now, after you get PS, essentially you get serine on top of the diacylglycerides, okay, diacylglycerol, whatever. All right, this is PS. First, membrane lipid, PS. Now, okay, now we're going to modify it a little bit. We say, PS, I don't like it, okay? I want this thing called PE, phosphatidylethynolamine, okay? Ethanolamine 是什么鬼 ？OK， ethanolamine 来哦，这是什么？这是 ethanol。ethanol 后面放一个 amine， 这种东西就叫做 ethanolamine。OK， ethanolamine。Right. Now, the reason why it's called phosphatidylethanolamine is because the OH is reacted with the phosphate, and then on the outside here, it's just 2 carbon and an amine. Okay. Now, how do you get that? You have a very simple decarboxylation here. This is a decarboxylase. Okay. All right. How do I know it's a decarboxylase? Because 它有 release 一个 CO2 嘛. Okay, simply. All right. So, okay, ideally, you should be somewhat comfortable at telling me what kind of enzyme can do this reaction. 可以吧？做 carboxylate 就是把 CO2 插在一个 molecule 身上的那种 enzyme 叫做什么 ？carboxylase。OK， 放 CO2 叫 carboxylase， 拔 CO2 的叫做 decarboxylase。OK， 然后我们有看到 dehydrogenase、kinase。OK， 所以throughout biochemistry， 我希望你们可以有时候看一个 reaction， 然后你就说、OK， 那这个应该是一个什么样子的 enzyme， 至少你叫得出它是一个什么 type 的 enzyme。OK， 然后你再去看它的 substrate 是什么，那 most likely 它的 name 就是 blah blah decarboxylase。OK， 这是最simple 的 way 去 communicate with biochemists. Okay, all right. So once your PS gets decarboxylated, you form this thing called PE, phosphatidylethanolamine. Essentially, you just lost the acid group of the serine and then gone, it becomes CO2, and then now you have phenylamine left, okay, and then PE. So as you can see on the bottom here, bacterial cell membranes are relatively simple, okay, in terms of their membrane composition. Okay, you have about phosphatidylethanolamine, so the PE portion counts for 70 to 80%, okay, and then you got the other stuff that goes at 20 to 30. Okay, so bacteria is very, very simple. Okay, you don't have all sorts of different kinds of membranes, but of course humans, we are a lot more complicated. Okay, so let's take a look. Okay, so you have seen this table in Biochemistry 1, like I assume you guys all forgot about it. Right? So in Biochem 1, we talked about membranes. In particular, we looked at these different types of membranes, and then we looked at different types of cell. So E. coli, a simple bacteria, you see is primarily PE. Right? So this guy's PE. And then you have some others. Okay, I'm not going to talk about what the others are. And then you have also a little bit of this phosphatidylglycerol. Okay, so you have glycerol reacting with it. Right? So bacteria, very simple. Okay, it's mostly PE, okay, for our discussion purposes. Now, if you look at higher level organisms and their compositions, and you see that mitochondria, you got PE, you got a little bit of PS, and then we'll talk about this thing called PC, phosphatidylcholine, choline or choline, whatever. Okay, 我念 choline 啦. All right, so phosphatidylcholine, it's about 40%, okay, in mitochondria, so it's quite large. So we're going to talk about that, okay. And this guy is derived from PE, okay. If you look at human myelin, okay, so myelins are the cells that's related to your neural cells, okay, like your nervous cells. And you got a bunch of glycolipids. We'll talk a little bit about glycolipids. And then cholesterol, okay, so we're going to talk about cholesterol biosynthesis, okay, but in lecture 14, okay, which I just uploaded the slides this morning, so you probably don't have it, okay. And then you also have human erythrocytes, plasma membrane. Remember, erythrocytes are red blood cells, okay? And you see that they also have different kinds of membrane structures, okay? Membrane lipid composition, okay? Now, so going from this chart, what do you know, okay? Now you know the synthesis of this guy, you know, okay? This guy, you know. Phosphatidylglycerol, I think you know, okay? This is this thing, okay? Before cardiolipin, right? And what else? Serine, you know? Okay, so those four, you already know where they come from, okay? 可以哈? Right? Now we're going to talk a little bit about the eukaryotic biosynthesis, right? Because choline, this guy right here, happens a lot in eukaryotic cells, right? So in mitochondria. So we're going to talk about how to get your PC, right? This is PE, and then this is PS, okay? All right, so since we're switching gears to eukaryotics, we're going to first talk a little bit about the biosynthesis of phosphatidic acid in eukaryotic cells, okay? So previously, when we talk about, you know, the middle part, right? 我们刚刚不是有看到那个 middle part, 那 middle part 那个是 eukaryotics 跟 prokaryotics 都可以用. Right? Now we're going to talk a little bit about other pathways in the eukaryotic biosynthesis. Okay, so what we just talked about was here. Okay, so this guy, a lot of people have. Okay, so bacteria, whatever, they all got this going from glycerol 3-phosphate. Okay, and then you get the first glycerol 3-phosphate, so one arm and then two arms. Okay, so this is what we just talked about. Now, in eukaryotic systems, you also got this other thing where you can go from DHAP. Okay, so you can go from directly at DHAP. You don't have to go from glycerol 3-phosphate. You can go from the dihydroxyacetone phosphate. What's the difference? Glycerol 呢这边已经是 OH 了 ，DHAP 呢这边还是 ketone。OK， 那记得 glycerol 3-phosphate 怎么来的？是这样子过来的。OK。那因为这边是一个 ketone， 这边是一个 OH， 那各位觉得我这边要吃一个什么？ 一个是 C double bond O， 然后变成是一个 C single bond O。ketone 变 alcohol 啊，很常看到啊。ketone 变 alcohol 怎么变 ？NADH， OK， NADH. So you do a simple reduction, NAD. I'm just going to call it NADPH because I don't know exactly if it's NADH or NADPH, okay? But it requires a reducing cofactor, 好吗？所以你只要看到说 molecule 它越变越 reduce, that means, okay, it's going to use NADH or NADPH, okay? How do I know a molecule is more reduced or more oxidized? You look at the number of oxygen, okay, that's the simplest way to look at it, okay? So the more oxygen you have on the molecule, it usually means that it's more oxidized. So the more oxygen you have on a molecule is more oxidized, right? That oxygen oxidized, okay? So it's easy to remember. All right, so in this case, you have on the right, DHAP has 2 bonds to oxygen at the C2 position. On the left, the glycerol has only 1 bond to oxygen at the C2 position. So you go from 2 oxygen to 1 oxygen. Okay, you can think of it that way, right? 这边是2个 O bond 在这个身上，然后这边是1个 O bond 在这个身上 ，right？ 那你就知道说1个 O bond 的比较 reduce， 2个 O bond 的比较 oxidize，OK？、Okay? 这个拜托要知道一下哦，因为这个太那个了，太基本了。Okay, so typically, of course, we go to glycerol 3-phosphate and then you make your diacylglycerides. But now, in eukaryotic systems, what I can do is I can have DHAP. Since I already have an OH here, what I can do is directly add a fatty acyl-CoA and then at first add 1 tail. Okay? Add 1个 tail 在这边。Right? 这边是1个 tail. Right? 所以我拿这一坨，然后这个 OH 不是会 attack 到 carbon 上去下来 ，CoA 不见。OK， 所以 always 如果你今天是要拿一个 alcohol 跟一个 activated fatty acid 去做 reaction 的话，它的 mechanism 都一定是这样嘛，因为你的 oxygen 身上有 lone pair。Lone pair attacks the carbonyl center, and then blah blah blah, and then you got this. Okay. Once you have the 1 acyl DHAP, okay, then you can reduce it. You still have to reduce the C2 position into an alcohol because otherwise you don't have a hydroxyl group to react with the activated fatty acids, okay? So in order to generate your final product as phosphatidic acid, you have to have 1 arm, 2 arms. And in order to have 2 arms, you need to have OH here, OH here. Okay, so you cannot just stay at ketone. You have to get it to alcohol, and then in order to react it with the acid, okay, because only alcohol can react with acid. 这个 what you may call it? Eukaryotic systems, okay, because we are eukaryotic systems, and what we can do is, guess what? You can eat lipids, okay? In your diet, 你可以吃很多 fat, okay? I don't recommend it, but you can, alright? So what you can do is you get this triacylglycerol, okay? 你吃进来，或者是你本来身体里面就有储存 ，okay? 你储存了一些 TAG， okay? 这是 TAG，okay? TAG 嘛，那 TAG 呢，它是长这样子。Right? All I gotta do, okay, all I gotta do is hydrolyze 1 out, and then it becomes phosphatidic acid. Okay, I mean, of course, you have to first hydrolyze it and activate it. Okay? So, if I take TAG, I hydrolyze 1 of the arm, or actually it has to be the top arm. You hydrolyze this guy, and you liberate the alcohol group. Right? And where's this O group, this OH group coming from? Is this. Okay, this O, right? Okay, so it's this guy, right? So you hydrolyze it, and then the oxygen is there, okay? And then you use what kind of enzyme would this be? It's going to be a kinase, right? 可以吧？因为它是用 ATP 嘛。ATP 然后把一个 phosphate 丢到一个东西身上，所以反正 some kind of kinase, okay? And then you activate your diacylglycerol into phosphatidic acid. Since we're interested in knowing how you make PC, okay, phosphatidylcholine. Okay, now choline is a very interesting molecule. Okay, the structure of choline is right here. So if I expand it out, it's going to look like this. Here's an OH, 1 carbon, 2 carbon, okay, and then you have nitrogen in the middle, and then what you get is methyl group, methyl group, methyl group, and now N is positively charged. And you're just like, whoa, okay, like, whoa, okay? Yeah, but it exists, and I'll tell you how to make it, okay? All right, so to get the phosphatidylcholine, okay, PC, right, PC, there are 2 pathways, okay? There are 2 types of pathway. One type of pathway is you make it from scratch, okay? 你从头去做 choline. Second type of pathway is you salvage it, okay? This is called the salvage pathway. 就是说，你今天从外面吃进来吃的东西，然后in what you eat, you got choline somewhere, okay? So you can salvage it. You don't have to synthesize it by yourself. You can just take choline and then put that onto phosphatidic acid and it becomes, you know, PC. Okay, huh? 来咯. Alright, so salvaging pathway, you have choline here. You first activate the OH group so that it becomes a phosphate. Okay? And then all you gotta do then is activate it again using CTP. Okay, it's a transferase. This is not a kinase, it's a transferase. On top here is a kinase. Alright, here you transferase, you transfer here a CMP onto phosphocholine, and then because here you already have a phosphate, you put on a CMP, together you get a CDP. CDP choline can then react with this thing DAG, is diacylglycerol, okay? Where you can then react from the phosphate to the OH. Okay, 那 reaction mechanism 还是一样哦，哈，还是一样。这边有 lone pair electron 嘛，对不对？然后它会飞过来 attack 这边，然后这个 bond 呢就会飞过来这边，然后 CDP 飞出去 ，and then 你的那个 choline 加 phosphate 就飞到 DAG 身上 ，and then you get PC, okay? And this exact mechanism you can also use it for PE. Okay, except all you gotta do is you have to start with ethanolamine. Right. So if you salvage ethanolamine inside of your growth media, and you can use this pathway to do it. The synthesis of phosphatidylcholine and phosphatidylethanolamine, they can actually interchange. Okay, so the second pathway to PE and PC actually starts with the conversion of PS to PE. Okay, so we saw that PE is derived from PS. 可以哈，你先有 PS 嘛，对不对？然后 PS 做一个 decarboxylation， 就会这边是 PS 嘛，哎，这是 PE， 写大一点 PS， 然后这是 PE。PS 到 PE 呢是一个 decarboxylation，OK， 所以你会踢出去一个 CO2。Okay, this we saw. This is phosphatidylserine decarboxylase converts PS to PE. And then you can actually convert PE to PC through this thing called the PE methyl transferase. I'll tell you what those are in just a minute. So we go from PS to PE, PE to PC. If we're talking about from scratch, 就是说从 linear pathway 就从头去做的话，就是 PS, P, PC. OK， 如果从头去做，但是你看这个 pathway, if you look at this pathway, you'll see that you got some interconversion thing going on. Okay, for example, 这个 PE 可以变回来 PS。仔细看哦，下面你用的可是 serine， 然后用一个 transferase， 把 serine 跟这边的 ethanolamine 做对调。Okay, 做 transferase， 做 transfer。So that you spit out ethanolamine, okay? But then you need to have serine coming from somewhere, okay? So this is not de novo, we're not synthesizing it from scratch. Here we are, okay? Here you have PS, and then you decarboxylate, it becomes PE, and then here you build. In this direction, PS, PE, PC, you're building, okay? Converting one molecule to another. However, the interconversion between, for example, PE to PS is a transferase. PC to PS is a transferase. Okay, so everybody can go back to PS. All you gotta do is put serine and you do a transferase. OK 啊，所以 PC 跟 PE 都回得去 PS。OK， 所以 just so that you know。OK。但是 PE 跟 PC 回去 PS 的方法是用 transferase， 而不是什么 carboxylase 啊，或者是什么一些 crazy 的 enzyme。We're going to look at this really cool enzyme, okay? This really cool enzyme, it converts PE to PC by methylating the ammonia group or the amino group 3 times using this enzyme called the phosphatidylethylamine methyltransferase, okay? Methyltransferase。Methyl group 啊，在 biology 里面，就至少在 metabolism 里面，其实不是很多。我们在 metabolism 里面最常看到的是 acyl transferase，OK， 就是 transfer acyl group 的。这边我们要 transfer 一个 methyl group，OK，now methyl group is it easy to transfer? No, right? So show you guys this thing. We're going to come back to this in more details when we talk about the synthesis of amino acids, OK？ 因为amino acid biosynthesis 我们会再看到这个，所以我今天先跟各位稍微介绍一下有这个东西，但是它的 exact mechanism 跟这个东西的 biosynthesis 我们到时候讲 amino acid 的时候会再跟你说一次。The methyl donor in most of biology, there are 2, and the biggest one is this thing called SAM, okay, or a lot of people will call it adomet, but I think SAM sounds more like 你跟它很 tight， 有没有？你跟它就是 SAM, okay, SAM is easy to remember, okay, just like the person's name. SAM stands for S-adenosylmethionine, so S-A-M, okay, SAM. Okay, but in your textbook, it's people going to refer to adomet. Met is for methionine, ado is for adenosyl. Okay, so either way people use, but I like to use SAM because SAM is easy to remember. Okay, now SAM is the methyl group donor in the synthesis of PC, okay, and numerous other methylated metabolites. Okay, it's actually used quite a lot. Okay? There are a lot of metabolites that you are trying to methylate. You will use SAM. Okay, now what is SAM? Okay, what does SAM look like? Okay, right? It's not just this random dude down the street called SAM, right? It's a molecule that is derived from methionine, right? All right, so methionine, do you guys remember what it looks like? Methionine looks like that. This is methionine. Okay, remember that the R group has 2 carbon, 1 carbon, 2 carbon, and then it has a sulfur atom in the middle, and then it's attached to a methyl group. That's what methionine looks like. Okay, now, from here, you should be like, oh, right, so methionine has a methyl group on the sulfur. Okay, so that's where it comes from, okay? So obviously then the biosynthesis of methionine requires methyl transfers. Okay, so we'll talk about that later. So SAM, all you gotta do is you take the adenosine moiety here. Again, ribose plus adenine. Right, so this thing is the same thing for when you want to make your like RNA and stuff. Okay, RNA, ATP, that kind of stuff. Okay, so it's all this thing. You put a adenosyl group on the methionine so that it looks like this. Okay? And then, this is SAM. Then you can use a methyltransferase. So these things typically called methyltransferase. Or sometimes it's going to be called methylase. It's weird. Methyltransferase, yeah. All right, so methyltransferase, now what it can do is you can then have some kind of a recipient, okay, some kind of recipient, and then you're going to throw this methyl group onto the recipient. So that's just all it is. Right? So you can do a very simple transfer, and you end up with SAH. So you go from SAM to SAH to like SA, okay, whatever. All right, so, okay, so SAM to SAH. Okay, H stands for homocysteine. Okay, homocysteine. 你会想说，哎，那跟 cysteine 什么关系 ？OK， 那个，你常常有时候听到 homo something。Homo whatever means that it has 1 extra carbon compared to the normal amino acid. So the normal cysteine would just have, one carbon and then thiol. But if you have homocysteine, you have 2 carbons here and then thiol. SAM biosynthesis starts from methionine, okay, obviously, okay. Now, if you start with methionine, and all you got to do is activate it using ATP, but this is not a kinase, guys. Kinase donates a phosphate. This guy actually donates the adenosine, okay, just the adenosine, okay. So this thing is called the methionine adenosyl transferase. So you are transferring the adenosyl group. This thing is the adenosyl group, okay, this whole thing, and then you transfer the adenosyl group onto methionine, okay, and the way it works is just like that, okay. So you're going to have some kind of a nucleophilic attack, and then you lose the phosphates, okay, the triphosphates, and then the triphosphates can then degrade to pyrophosphate. Pyrophosphate goes to phosphate. So all of these delta G is pretty less than 0. And once you get to this SAM, okay, and then you can use it to do your methylation reaction. Now, here it says that the triphosphate is hydrolyzed by the enzyme to pyrophosphate, pyrophosphate to phosphate, okay, and this hydrolysis, because every step here the delta G is less than 0, so it drives this reaction, okay, 因为你不能再把 triphosphate 塞回去, okay, now triphosphate goes to pyrophosphate, pyrophosphate goes to monophosphate, okay, right, so this is how you make SAM. Okay, so we talked about PS, PE, PC, okay, and the next thing we're going to talk about is this thing called PI. I'm not talking about the isoelectric point. Here it means the phosphatidyl inositol, okay, it's a type of sugar, okay, it's a sugar alcohol, right, this is whatever tol, okay, it's sugar alcohol, right? And the way it works, okay, you have to first make your inositol here. You make your sugar first, okay? And then what you can do is you take the CDP diacylglycerol. Remember where this is from, right? This guy is right before PS, right? So if you take the CDP diacylglycerol, you can use that to make PS. You can use that to make CDP glycerol, okay, that kind of stuff. But here, what we're gonna do is to react it with a sugar alcohol to be exact, okay? This guy is kind of like a little sugar alcohol. And then what we can do then is react it also using one of the OH with the phosphate, okay? Just like how your PS works. OH attacks the phosphor, and then, CDP gets kicked out, okay? And then you get this thing called the phosphatidyl inositol. So here you have a little sugar here, okay? And then what you can then further activate it to is you can start adding charges, okay? Let's say you want to have a lot of negative charge on your membrane, okay? So then you can keep on using these kinases to then phosphorylate all these OHs such that they become O-phosphate, okay? So then they will become negatively charged, right? Okay, so this is just a type of membrane lipid. Now, what's also important for us is that PI, this thing, is often participating in linkages used for glycoproteins. Okay, what does that mean? PI, this thing is a lipid. Okay, so it's going to be inside of the membrane. Okay, but then what if you want to put a protein on the membrane? Okay, and if this protein is a glycoprotein, 它是有糖，上面有糖的 protein， 那它就可以借由这个 linkage 去把你的这个 protein 呢插在 membrane 上。OK， 所以它是一种机制来把那个 protein 放在 membrane 上面。OK， through this glycolytic linkage. Now this is alkyl ether. Okay, not ester, ether phospholipids. Just so that you know it exists. Okay. sometimes, okay, you're going to get this really weird stuff, okay, really, really weird. What this is you first make the one-tailed dihydroxy acetone phosphate, okay, so remember you go from DHAP and then you can add one fatty acyl ACP, all right, we'll CoA, whatever, and it becomes this, right? Now, interestingly, okay, what you can do is you can swap it using a fatty alcohol, okay? How do you make a fatty alcohol? You start with a fatty acyl CoA, you reduce it twice, okay, so you go from CoA, first reduction, you go down to fatty acyl CoA fatty acyl CoA 它是一个 fatty thiol ester， 对，它是一个 ester。那 ester 它的 oxidation state 跟什么一样？跟 acid 一样，跟 organic acid， 跟 carboxylic acid 一样，对吧？哈，那 carboxylic acid 做一步还原会变成什么 ？aldehydes， OK， 会变 aldehyde。Okay, so this guy become an aldehyde。然后你再做一步的 reduction， then it becomes alcohol. That's why 2 NADPH. Okay, one for this guy, one for this guy. Okay, so you generate this fatty alcohol, and then what this fatty alcohol can do is you can then substitute it with the fatty acids. Okay, on here, such that you generate this thing. This is the ether linkage. 就是 blah 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 C O C blah 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 blah. OK， 这种东西。这个东西记得是一个 ether. Yeah, doesn't happen a lot, but it's there, just so that you know it's there. Okay, and then what you can then do is here initially it's ketone, right? DHAP. You reduce it into alcohol, and then you add a normal fatty acid, and then it becomes this. It still has 2 arms, right? 2 tails, but it's just that one tail becomes ether linkage instead of ester linkage. OK， 好了，你想说来这个东西到底有什么用？ But 就跟你说一下它 exists 嘛，好不好？ Just so that you know it's there. Okay, maybe it's harder to degrade. Esters are very easy to hydrolyze. Ethers maybe slightly harder. Okay, so we finished talking about phospholipids, right? So hopefully you guys now know how to make different phospholipids. Now we're going to spend a little bit of time on sphingomyelin. It's important. Okay, why it's important? I'll show you towards the end. Okay, so there's a lot of hereditary diseases. Okay, human diseases are related to sphingomyelin metabolism. Okay, so if you cannot properly degrade sphingomyelin or synthesize sphingomyelin, it's very easily that you can get some problems. Okay, some diseases. But that's true for everything, right? All right. Myelins are, first of all, myelins. So this is your axon. So axon is like going this way. And myelins are the layers surrounding the axon. They serve as insulating layer, okay? Because, I mean, come on, axons are, they send electric pulses and stuff like that. So obviously you're going to want to insulate them because otherwise you're going to have problems. Right. Now, many diseases are related to the genetic defects of sphingolipid metabolism, turns out. Okay, so we'll talk a little bit about that, but not so much. So in humans, sphingolipids constitutes about 25%, slightly more, slightly less, about total lipids. So it's actually quite large in humans. Okay, so 25 lipids, so 1/4 of your total lipid are made out of sphingolipids. Okay. So in a normal individual, sphingolipids are in continuous state of metabolic turnover. Okay, so this is an interesting concept. Metabolic turnover meaning both you're synthesizing it and degrading it simultaneously. So you're always making new sphingolipids. Okay, just continuously making new sphingolipids. Therefore, because you're always making new ones and degrading the old ones, making the new ones, degrading the old ones. So as a result, a lot of these metabolic diseases, okay, there's a name for it called sphingolipidosis. They're related to deficiency of the degradation enzyme. So if you cannot accurately and appropriately degrade sphingolipids, but you're continuously making them. So you keep on making these sphingolipids, but you're not degrading them. As a result, you get some metabolic disease. As sphingolipids are often related to nervous systems, so sphingolipidosis are often very devastating. Devastating meaning it's really bad, okay, because it affects your entire motor, sensory, all types of different functions in your body related to nervous system. So quite a lot. All right, so a brief reminder of what sphingolipids look like. You can go to as simple as this thing called the sphingosine or sphinganing. We'll see that in just a minute. All right, so this is the basic structure of sphingolipids, okay? So there's one lipid arm. Here's the amino group and two hydroxyl group. I know you guys can't see it from the back, but if you go download the slides, you can see it. If you put fatty acid residue on the amino group, then you get a ceramide. Okay, so ceramide now has two legs, okay, because it has one already from the sphingosine, and then you have another fatty acid residue. So ceramide constitutes a functional membrane lipid. Okay, so two arms, two tails, whatever. All right, two tails, and hydrophilic groups. Okay. And then you can further take ceramide and you make it into sphingomyelin. So if you look at the structure between ceramide and sphingomyelin, all you see is you have your favorite phosphocholine group. Right? So your phosphocholine. And then going from that, you can also, same thing, you can have ethanolamine, phosphoethanolamine. So these two are they're there. So as you can see, that choline, ethanolamine, they're prevalent on membrane lipid. And then you can also generate cerebrosides. Cerebrosides are using sugar, one simple sugar, okay, one sugar on the first hydroxyl group there. Or you take cerebrosides and you start to hook up additional sugar moieties, different sugar moieties, then you get this thing called ganglioside. 它们反正就是不同的 sphingomyelin lipids, okay? Sphingomyelin lipids. Sphingolipids, all right? So we start with this thing called sphinganing or sphingosine, okay, sphingosine. So first, glycosphingolipids, okay? How do we make them? Okay, glycosphingolipids comes from using ceramide plus sugar at C1, okay, what we just saw, all right? So cerebrosides typically just one sugar, and they are typically glucose or galactose. Right, that's cerebroside. Gangliosides are a lot more. Okay, so you have different numbers of sugars at different positions, but what you notice is that on gangliosides you have this thing called the sialic acid. So it's a sugar acid. Okay, so sialic acid. Minor detail. And these molecules are especially common on membranes of nervous cells. Okay, so how do we make sphingolipids? We have to first generate this basic structure, okay? This basic structure. The way to do that would be you take serine, again, your best friend serine, plus a C16-CoA, okay? So palmitoyl-CoA, 16 carbons, okay? So essentially it's a 16 carbon fatty acid, but activated. All right, and then you add them together, okay? And you'll get this thing called the 3-keto sphinganing. Okay, so where do you add them? You add right here. You add it to this carbon. Okay, so CO2 is lost. Okay? CO2 is lost, and then you essentially replace this CO2 with your fatty acyl group. And then after you get 3-keto, okay, 3-keto because the keto is right here, and this keto group comes from this guy. The 3-keto here, and then all you have to do is reduce it using some kind of dehydrogenase, and then you get sphinganing. And then sphinganing can then, since you have an amino group right here, you can react it with another fatty acyl-CoA. This guy is a fatty acyl CoA, and then you get ceramide. Just like in phospholipids, you take an activated fatty acid, okay, fatty acyl-CoA, you put it on to glycerol 3-phosphate, same idea. But here you are putting it on to a amino group, okay? Amino group, just amino group, okay? So ceramide, once you get ceramide, you will actually go from the ceramide here, it's single, all of them are saturated, and then you can do a desaturation, okay, in animals. And then you generate this ceramide, but with alkene inside. Okay, I don't know why they're both called ceramides, but It's just their naming. And then you go from the ceramide, you can then start adding different things. If you add choline through phosphatidylcholine, you transfer the choline portion from the phosphatidylcholine to the ceramide, then you get sphingomyelin, because here this is phosphocholine, okay? And your PC then becomes your diacylglycerol because it passed the choline portion onto ceramide. Make sense? 有听懂我意思吗？就是你从我们刚刚的这个 C18 的 fatty acyl CoA 加上一个 serine 之后，会先变成这个 3-keto sphingane， 然后我们把 keto 先 reduce 掉 ，OK， 它就会变成 OH。那变成 OH 之后呢，这个其实就很像一般我们 fatty acid 的 structure 了，因为它有一条很长的 lipid arm， 然后这边有一些 head group，OK， 然后 sphingane 呢会先加一个这个 fatty acyl CoA， 然后我们加在哪里 ？OK， 我们 always 加在这个 C2， 因为两个 lipid arm 要在一起，然后那个 polar arm 往上 ，OK， 所以你就会形成这边是一个 fatty arm， 然后第二个 fatty arm，OK， 两个 tail， 两个 lipid tail， 然后你就有这个东西叫 ceramide，OK， 然后 ceramide 呢之后，如果我要把它变成这个 sphingomyelin，OK， 那我就可以拿我 PC 上面的那个 C 有没有跟它就是把它 transfer， 把那个 C transfer 到我的 ceramide 身上，那它就会变成我的 sphingomyelin。那这个时候 PC 呢就会变成 diacylglycerol。OK， 那记不记得我们刚刚有讲到说 eukaryotic cells， 我可以再重复利用我的 diacylglycerol，right， 再去做 PC。Or what you can also do is then you take ceramide and you take UDP glucose. Do you guys remember where we saw UDP glucose? 有没有记得？ We saw UDP glucose somewhere when we talked about the metabolism of like galactose metabolism, right? We saw, in order to use galactose, you have to use UDP glucose and transfer and all that stuff. Okay, so UDP glucose is not a uncommon metabolite, so it's actually used in many places. Right? Using UDP glucose, you can pass the glucose moiety onto the top OH right here, and then you generate cerebroside. And going from cerebroside or ceramides in general, what you can generate, let's say this is ceramide, the basic structure right here, basic structure. And you can generate all kinds of different sphingolipids, glycosphingolipids. And to do that, all you have to do is keep on using these UDP glucose, UDP galactose, these UDP sugars, and then they can keep on adding sugars onto the chain such that you can generate, for example, ceramide with a galactose, ceramide with a galactose, plus another galactose. Or if you get more complicated, you can get ceramide with a galactose, glucose, galactose, sialic acid. Okay, so you can keep on adding these different sugars onto your ceramide, and you form all these different gangliosides. Okay, so these things are the gangliosides. The degradation, then let's say you build all of these. Now you have to degrade them. How do you degrade them? So if you want to degrade them, so you have these glycosidic bonds, right? Glycosidic bonds. All you got to do then is to hydrolyze them. Okay, just like when we talked about back in glycogen or sugar chemistry. Okay, so you do a simple glycosidase type of reaction, and then you will be able to hydrolyze the glycosidic bonds. Okay, right? So that's how you synthesize and you degrade ganglioside. Now, all the different kinds of genetic diseases that are related to sphingolipid catabolism are listed here. Okay, so there are different types of diseases as you can see, and if you have defects in all these, for example, glycosidase, acetohexoaminosidase, galactosidase, beta-glucosidase, okay, all these enzymes that are related to the degradation of sphingolipids, if you have defects on those enzymes, then you will accumulate. Okay, you will accumulate different type of sphingolipids. For example, Some of them you accumulate sphingomyelin, some of it you accumulate ceramide, some of it you accumulate gangliosides, okay? And as a result, you accumulate them, you'll have, they will mess up your nervous system because you accumulate a large abnormal amount of these lipids that are related to nervous cells, okay? Now, for my purpose, you should understand why they accumulate. Okay, that should be simple, right? You should be able to understand why is it that if you have defects on the degradation genes, they accumulate. Okay.